Welcome back here. Now that we've kind of talked about what we want to say concerning the cell communication, I wanted to make sure that we took a look at the cell cycle next, um, because the cell cycle is going to be really informative about how we make new cells from old cells. Um, we're going to spend two parts here on the cell cycle. Um, First, today we're just going to spend some time setting up information. So, setting up some key terms and vocabulary words before we look at the parts of the cell cycle tomorrow. And then we're going to kind of finish up about the cell cycle next week when we're going to look at how the cell cycle is regulated. And then we'll take a look at that next week. First, we gotta kind of want to know um, why the cell cycle is important. And the cell cycle leads to, as part of the cycle, the division of cells. So making new cells from old cells, the continuity of life. This is how cells reproduce. All right, how they make new cells. And if you think about organisms that are only one cell big, that's how they multiply. Instead of going through sexual reproduction, they go through asexual reproduction where they just duplicate themselves, right? But a lot of what we're going to talk about has to do more specifically with multicellular organisms like ourselves and plants and fungi and all of those cool things. And a lot of the times, cells need to divide in order to allow the multi-cellular organism they're part of to grow, so they get bigger, to repair damaged cells and to renew ones that are really old, all right, or to make new tissue. So, for example, when you might go on an exercise regimen and try to gain muscle, those cells, those muscle cells, would need to divide more frequently to produce new, more strong muscle cells. When we're talking about cell division specifically as part of the cell cycle, because the cell cycle isn't just dividing, we're looking at how we make two new cells from one old cell. All right, so that's kind of the long game where we're going to want to be um, eventually. But for now, we want to set up some building blocks. Because if you think about it, if you're going to divide a cell, you are going to need to make sure that you have double the DNA that the original cell has, so that when it divides and you have two new cells, they each have a full complement of DNA and not just half of it. So we need to talk a little bit about DNA first, a little bit more information than we got in the biochemistry unit, just to make sure that we know what's going on when we're duplicating our DNA. And we're gonna wait and talk about duplicating DNA until we get to genetics. But just the basics is that DNA, that double helix, is organized into a chromosome, kind of like you see down here, just a long cylindrical packaging of the DNA double helix. And that DNA double helix is actually wrapped around proteins called histones, kind of like these histones. Okay, they're kind of wrapped around it to pack them tighter together because each of your cells has about a meter of DNA if you just stretch it all out. And the only way really to get that to fit into the nucleus of a really, really small cell is to wrap it up really, really tight, and those histones help with that, right? You can see here kind of arrangements, a pattern of 8 histones kind of grouped together, eight more, eight more. We call that kind of a nucleosome. So making a little bit bigger unit we can refer to out of these smaller units. So when DNA is in this state, with the DNA double helix wrapped around these histones, we call it chromatin, that long fibrous, mesh string of DNA. So here's a picture kind of showing you a really small view, close up, the nucleosomes, and then the large view of the whole chromosome. So a chromosome itself is the megastructure, the big structure of where DNA is stored. And an important thing to realize is this X shape that you might find analogous to a chromosome is only present when the DNA has been duplicated. So this is actually a duplicated chromosome. Before they're duplicated through much of the cell cycle, they just look like this one up here, right up here. Just, oops, a long kind of cylinder. And then when the DNA duplicates, it turns into this X shape. That means that each half of this X shape is an exact copy of the other half. And so we call one half of this X shape chromosome a chromatid. And these two are sister chromatids, right? And you might notice that there's this small part in the middle where they're joined or stuck together. We call that the centromere. So the centromere joins the two sister chromatids together. Each of them is an exact copy of the other, one duplicated chromosome. And then the last thing is these very ends, like up here, and down here on both sides of the chromosome are called telomeres. And they contain long repeated, basically nonsense segments of DNA that serve to protect the rest of it that has genes and important information in it from being degraded. So the idea is that if this DNA would start to wear away over time, it would do so at the ends first, and the parts that were being weared away were these telomeres that didn't really contain any important information anyway. Um, Telomeres have been really linked to aging, and one of the things that science is looking into is whether much of the aging process or the negative effects of advanced age come from these telomeres from wearing way, way, way down because they get a little shorter every time a cell divides and makes a new cell. So the longer the multicellular organism is dividing or alive, the smaller the telomeres will get, and the closer to the DNA that actually contains valuable information will it get. So here's another picture of an unduplicated chromosome. So this is one that's not been duplicated yet. You can see the telomeres in yellow. These bands just represent different regions of the chromosome, and here in the middle is the centromere. Um, just remember that all of what we talked about there had to do with eukaryotic cells. So prokaryotic cells contain much of their DNA just in one big loop called a plasmoid. And then they have a few other genes on an accessory loop called a plasmid, which we'll talk more about in genetics and genetic engineering. But the chromosome structure and the histones aren't the same for prokaryotes. So just a reminder as far as that goes. So in general, in most multicellular organisms, sorry, there are two types of cells. called somatic cells and gametes. Somatic cells make up 99% of your cells. They're like the body cells, like a brain cell or a muscle cell, a heart cell, a liver cell, a skin cell, all the different types that make up most of your body. In those somatic cells, as far as humans, we have 46 chromosomes. We have two of each. So we have 23, no, sorry, 23 chromosomes, but we have 23 pairs of chromosomes. So we have two copies of each chromosome. We say that they're copies not because they're exactly the same, but that each chromosome in a pair contains the genetic information for the same gene or the same trait. Okay? They might not have this exact same DNA on them, but they have the same genes on them. And we call cells that have two copies of each, or pairs, diploid or diploid. So all of our somatic cells as multicellular organisms have diploid number. Sometimes write it as 2n. That means they have 46 chromosomes for us. Another organism like a cactus might have their somatic cells might have a diploid number of 84. All right, cacti actually do have many more sets of chromosomes than we do, and we'll talk more about that again in genetics. But if they have a diploid number, that just means they have two of each chromosome, that their chromosomes come in pairs. Gametes, on the other, the sex cells like the sperm and egg cells, only contain 23 chromosomes in humans. So we say that they're haploid. That means that they only have one chromosome from each pair, right? Like, so if we would number the pairs, chromosome pair 1, chromosome pair 2, and that's what we do, they only have one chromosome out of the two from each pair. So half the DNA. Haploid kind of sounds like half, okay? That's kind of how you can remember. This is why when the sperm cell and the egg cell meet during fertilization and form a zygote, which is the very beginning of new life, the 23 from the sperm cell, one chromosome from each pair, and the 23 from the egg cell, one from each pair, can form together to form a zygote that is diploid, that has two chromosomes from each pair, one from the uh, mother and one from the father. Right, and that's why we can have a new combination of traits in the offspring. Okay, so the gametes, the sperm and the egg cells, would be haploid, and the zygote, the new life, would be diploid because it has two of each chromosome instead of one of each chromosome. Right? When we're talking about um, diploid and haploid and pairs of chromosomes, when we talk about the two chromosomes in a pair, so like I said, we have chromosome pair 1, we refer to the two chromosomes in that pair as homologous chromosomes or a homologous pair. They're both chromosome 1s. They both belong in a pair together. They contain the same type, but not exact copies of genetic information. So each of the chromosomes in the homologous pair contain the same genes. They might not contain the same genetic information about those genes, but they contain the same genes, right? When, like we'll talk about mitosis, we go through DNA replication. Each of these chromosomes from the homologous pair reproduces or copies itself, replicates. And so now each chromosome from our homologous pair looks like an X, and they each have two sister chromatids, just like this. Like I said, just wanting to get some terms out of the way first today. Today's all about terms so that we can use them later, and we all know what we're talking about. So here's another example of two uh, homologous chromosomes, and these bands represent maybe different genes, and just pointing out, again, that idea that the homologous pairs of chromosomes have the same genes on them. They have genetic information that codes for the same traits, even though the exact bases over here aren't exact copies of over here. So, I think as far as what we're going to do for today, I want to stop right here because that gets a lot of the terms out of the way. So then we can use these terms when we're describing the cell cycle tomorrow.